for the here and now, we're going to focus in chapter 1 on the genetics of heritability and gene expression. Now, the one I'm showing you in the background, that this, a little bit of a chart, let me get rid of myself here. This basically is a chart that I've included genetic mutations in disease. When and where mutations occur indeed matters significantly. On the bottom here, you can see my cursor. Generally, when we talk about mutations, we're talking about mutations that occur in the cells that give rise to gametes. So we're talking about inheritance, we're talking about heritability or heredity, and ultimately changes that occur in the progression toward the generation of a gamete are those that occupy the concepts of population genetics, and ultimately we're the topic of things like evolutionary genetics, biology 427, human genetics, biology 460, etc. In this case, we're over here. We're looking at somatic mutations that occur in a particular cell, in a particular target tissue and organ, that is influenced by the environment. And it's these mutations that go on to ultimately give rise to the cancer phenotype and highlight the many changes that are required, generally about 5 or more somatic mutations in all of their myriad forms that go on and give rise to the primary tumor and ultimately to the metastases that result in secondary tumor formations. That's the concept that I really wanted to hit home. I'm going to go back to my shared screen and find this chapter's figures. Let me open this up, and I've basically distilled a few of the figures. We'll go back to the title of the chapter. This is the first figure that I would like to show, and this is a normal karyotype. You can see here that chromosomes are numbered, human chromosomes. We have 22 pairs of autosomes. They begin numbering with chromosome 1 being the largest, all the way down to the 22nd autosome, the smallest. And in the bottom right, you can see the X and Y chromosomes that constitute, in this case, a male karyotype. What is presented here is what's known as a fluorescence in situ hybridization experiment, in which various probes that are derived from chromosomes, we call these chromosome-specific probes that are involved in coloring the individual chromosomes, because they actually contain a unique fluorescent marker that's converted by a computer into these beautiful colors. You can see that those specific chromosome probes are giving rise to a very nice painted set of metaphase chromosomes that give rise to this beautiful karyotype. This is what is a normal karyotype. Now, in the cancer state, we're talking somatic mutations that give rise to gene expression variation. And we can look here at the same sort of probes applied to a particular cancer type in which we see a lot of change. Notice the breakup of the chromosomal integrity. Chromosome 1s, we have a number of yellow elements that mark chromosome 1 specifically, but now they've been attached to other elements because of end joining. We see here that not only do we have these strange rearrangements, probably due to end joining and chromosome breakage, but we have the generation of ploidy differences. In this case, aneuploidy. We have a situation here of trisomy chromosome 2. Here is a trisomy 16 with also a translocation, which part of 1 has ultimately moved onto and fused with chromosome 16. So you can see here we have a lot of changes. Monosomy chromosome 22 or monosomy 22. In addition to the retention of the X, but it appears that we've lost the chromosome Y. So we have a lot of genetic change, karyotypic change, that is quite indicative of the cancer genotypic nuclear state. So megabase changes, karyotypic changes, chromosomal changes that ultimately reflect the chaos inherent within a cancer cell. And this basically reflects the genetic instability that accompanies the progression from a normal cell to a full-blown neoplasm or a malignantly transformed cell that oftentimes biologists and oncologists just call a tumor, which doesn't really tell you whether it's benign, contained, or whether it has the ability to slough cells that move throughout the body and establish secondary homes, secondary tumors at distal